Hello and welcome to the Central Innovation and Methods Seminar Series. I'm Shirley Wang from Brigham and Women's Hospital in Boston, and I'll be moderating today's session. The U.S. Food and Drug Administration's Sentinel system helps to address questions related to the safety and effectiveness of marketed medical products. The Sentinel Innovation Center focuses on developing innovative methods to further advance Sentinel by leveraging new data sources, building new tools, and enhancing real-world evidence capabilities through advanced analytics. The Innovation Center works closely with the Sentinel Operations Center, which focuses on supporting FDA in the use of Sentinel Common Data Model and associated modular programs or tools to gather evidence to support regulatory decision-making. The Sentinel Operations Center maintains FDA's Active Risk Identification and Analysis System, known as ARIA. We launched the webinar series to engage experts and innovators in key areas related to the Innovation Center's master plan and the Operations Center's methods work. With that, I am pleased to introduce today's speakers, Dr. Judy Marrow and Dr. Joshua Smith, with whom I co-lead the newly formed Signal Detection Core of the Innovation Center. Dr. Marrow is an assistant professor in the Department of Population Medicine at Harvard Medical School and the Harvard Pilgrim Healthcare Institute. Her main research interest is in the implementation of pharmacovigilance techniques, particularly continuous near real-time sequential statistical analysis methods and data mining and signal identification methods in distributed longitudinal databases. She's also the operations lead for the Sentinel Operations Center. Dr. Smith is an assistant professor in the Department of Biomedical Informatics at Vanderbilt University Medical Center and a member of the Center for Precision Medicine and the Center for Patient and Professional Advocacy. His primary research focus is on the use of natural language processing of unstructured clinical documents to enable clinical decision support, phenotyping, quality of care improvement, and adverse drug effect detection and discovery. The title of their presentation is Introduction to the Signal Detection's Analytics Core in the Sentinel Innovation Center. Before we get started, I'd like to mention that your lines are muted. If you'd like to pose a question, please type it into the Zoom question and answer feature. We will answer questions at the end of the session. Finally, this session is being recorded and will be made available through the Sentinel webpage, which can be found at sentinelinitiative.org. Without further ado, Judy and Joshua, thank you so much for joining us. I'll turn it over to you.
Thanks, Shirley. Hi, folks. Welcome to our webinar today. I want to thank everybody for giving us the opportunity to speak and to talk with you all about this new exciting core within the Innovation Center. So I wanted to start out my remarks today with just a little bit about what we mean when we say signal detection, sometimes also called signal identification or signal generation. So when the Food and Drug Administration is looking at particular medical product exposures and then looking to see if there are high levels of outcomes that follow exposures, they're looking to see if there's something specific that's of interest. And so when there's a prior hypothesis, we sometimes think of that as signal evaluation. But when there's none, when we're not really sure what exposure outcome pairings are needed or something that we need to look at, we think of that as signal detection or identification. And so routinely at the Food and Drug Administration, the way that signal identification is performed is by looking at what's known as case reports in the FDA's Adverse Event Reporting System, which is also abbreviated FAERS. Additionally, there are case reports that come in through published medical literature. So these are typically published in medical journals. And so there's a team of folks that review these individual reports and articles, and they also perform analyses on them to look at which particular reports are indicating that a particular exposure outcome pairing is occurring more often than what would be expected based on the total volume. And so these techniques are the way that FDA sifts through the vast number of reports that they get and chooses which reports to investigate and which things to look at to try to understand more about what outcomes might be happening after those exposures. And these analyses that are then performed take into account all of the information available to the FDA at the time. So they include reviews of the reports in the FAERS system, they include reviews of the medical literature, they include reviews of the reports that are received from the manufacturer. Basically, they use all available information to them to understand more about a particular exposure outcome pairing. And that process is done at a risk-based approach. And so there's a reference there in the slides that you see with respect to how they do that. And so if you're more interested in the specifics of that, I encourage you to go to that reference. Next slide, please. So I want to point out that this system has worked tremendously for quite a long time. FAERS has been a key source leading to regulatory action. And what I mean by regulatory action is making a regulatory determination, which in this particular article that I'm showing you is an example of one, is a drug safety communication, where particularly the FAERS system contributed a majority of the time to when there was drug safety communication issued. Next bullet, thanks. Another paper shows that the number of safety-related drug labeling changes were often prompted by things that were first seen in FAERS or in the spontaneous reporting system. So the bottom line is that this has been a reliable source of information for the FDA for quite some time. Next slide, please. So FAERS has some known advantages and disadvantages, and they are very well studied. I'd encourage you to go to PubMed. There are many reviews of this type of information. But to summarize, some of the advantages are that FAERS is really good at detecting rare and acute events, things that happen very close in time to when a particular exposure happens, and things that have a particular drug-related signature. Those FAERS reports can be filed for all products and in all settings of use. And often they have detailed information about a particular patient. So they're coming from patient perspective. They have lots of very rich patient information sometimes. Some of the disadvantages are that there's an unknown denominator. So what that means is if you're exposed to a particular medical product and then you have an outcome, an obvious question will be, well, of all the people exposed to this product, how many of them have that outcome? And that's unknown in the FAERS system. Additionally, there is known underreporting. So one of the comments about a spontaneous report is that someone has to hypothesize that a particular exposure is related to the outcome. And if they don't see that connection potentially, it's going to be underreported. And sometimes there's also things known as stimulated reporting. So this can happen when, for example, an influential person reports that they have an outcome after having been on a particular product. And there's been documented instances where then you see more reports of that nature coming into the FAERS system. Additionally, the reports themselves, while they can contain rich information about patients, sometimes are missing key pieces of information. So in general, there are times where we know that FAERS is sort of not our best option for certain things. So anything that has a really long latency period. So if it takes several years, for example, for an outcome to develop that is related to an exposure, it's going to be more difficult for the FAERS system to detect that. If there are outcomes that have very high background rates in the population, if you can go back one more slide, the high background rates relate to for example, something like cardiovascular disease. Because it's so prominent and prevalent in the population, it's going to be hard to connect that particular disease to a particular medical product exposure, or any time where there's really not a good suspicion about what the causal mechanism is. And then back to the lack of denominator, you can't quantify the actual risk because of not knowing of all the people who are exposed to this product, how many experience this outcome. So now, next slide, please. Long ago, people looked at Sentinel as an opportunity because now here we have longitudinal data. We have the possibility of doing something that was known as active surveillance as opposed to what spontaneous reports are sometimes called passive surveillance. And so here with longitudinal data, we have the possibility of getting at that denominator. So we can say of all the people that we have exposed to this product, how many had this particular outcome? And then additionally, next bullet, please. We have the ability to control more richly for confounding information that's available in a person's longitudinal record because it has a lot of their history there. So are they somebody who is in a higher risk subgroup, for example? A lot of that information is available in a longitudinal database system. Next bullet, please. And so additionally, there's support for this idea of active surveillance first began in what was then known as the Institute of Medicine. Now it's called the National Academies of Medicine, but they had a report in 2007 called The Future of Drug Safety. A lot of this information eventually came into law as the 2007 FDA Amendments Act. And I've excerpted a portion of the act that you can see there, which basically is the birth of the Sentinel system as a way to look for particular outcomes that happen after exposure to particular medical products, and to do so not only for signal evaluation, but also for signal detection. Next slide, please. And so to compare these two systems, again, a longitudinal system like Sentinel compared to an adverse event reporting system, some things they have in common. As I mentioned before, in both cases in a signal detection framework or a signal identification framework, there's no need to say a particular exposure outcome pair you're interested in. It's intended to be a general safety net, and it's intended to produce hypotheses. And so next bullet, please, is about hypothesis generation. So the point of this, right? And the idea here is that if something rises to the point where you want to investigate it further, that's the whole purpose of these types of systems. Next slide, please. And so the data themselves in both of these systems have a hierarchical structure to them. So you can look at clinically related groupings of outcomes. And so that's a benefit of both of these systems. But there are some important differences. And so the first difference is the actual data source itself. And so again, the longitudinal data sources are compatible with a variety of familiar epi designs. They take advantage of that longitudinality, and they allow you to do an analysis in a way that's not really feasible in the FAIR system. Next bullet, please. The analytic data sets themselves are separate because the analytic data sets in a longitudinal system are looking more at population-based information. And so they're relying on these summary level data sets as opposed to the patient level that's available in the FAIR's case reports. They have different multiplicity control, and so that relates to formal multiple hypothesis testing, so looking at many different outcomes after exposure, and they have different comparison groups. And so again, the Sentinel system with the epidemiological focus is selecting a particular comparison group to look at, whereas the FAIR system is looking at all spontaneous reports as its comparison group. So next slide, please. So the signal identification availability within the Sentinel system, we start out by thinking about what design do I want to use, which really boils down to what kind of comparison do I want to have. And so if you look over at the rows there, the study designs include a self-controlled design where I look for information about a particular person and I compare two periods of that person's lifetime. And so in one period, they're exposed to a product, and in another period, I consider them unexposed to a product. I also could look at different kinds of cohort designs. Some of those cohort designs can involve matching either on exact characteristics like sex or age, or they can involve matching on a score. So a propensity score is the most common sort of use for matching there. Additionally, I could look at a stratified design where I basically break up my cohort into particular groupings based on either, again, exact characteristics, or there could be strata that are created based on propensity score quantiles. And each of these designs is compatible with certain types of longitudinal signal identification methods. And these are some of them that are available in the Sentinel system. And you'll see that a lot of them are available with a method that's called TreeScan, which has been heavily developed within the Sentinel system for use in looking at different exposures and all of the outcomes associated with them. Next slide, please. So here I'm just going to go through, there's some published literature so you can keep advancing. Megan, there's a couple of, essentially this is for folks who are able to go back to these slides when we get them posted on the internet and you want to go ahead and you want to look at references. Here are a bunch of references that use those self-controlled designs in particular, and they use a signal identification method known as the tree temporal scan statistic. Next slide, please. The next type of designs are the propensity score match designs. So actually, and Shirley, who so kindly introduced us, is one of the lead on this type of work. And she has a paper that we hoped is going to hit this second piece that you're seeing here is a presentation that should hit as a manuscript in the next week or 2 weeks to PubMed. So keep on the lookout for that. And then the next piece are the stratified cohort designs with a referent cohort. And so this was where a lot of initial work was done with research, looking at these designs. And now they've seen a resurgence as we've started to look at doing signal identification among pregnant women, looking for birth outcomes. And so if you advance the slides, Shirley has been one of the leads on this as well. And a paper that she's just published is newly in the literature, looking at active surveillance of the safety of medications used during pregnancy. All of these represent sort of the pathway of different TreeScan methods that have been used with data that is longitudinal in nature, like the Sentinel system. Next slide, please. And so that's sort of a review very briefly of where we've been. So what the past has been, what we're currently doing. But the whole point of the Innovation Center is to look ahead, and particularly to look ahead to the use of structured and unstructured electronic healthcare record data and how that can be incorporated. Next slide, please. And so recently, I mean this week, the Innovation Center Master Plan has been published. And so you can go, there's a reference there in the bottom of the slides. You can go there and take a look at this master plan. And the master plan has a particular core that we're talking about today, the signal detection or the detection analytics core. And in that core, the whole concept is to think about how can we take these approaches that have been used in the spontaneous reporting system and also the longitudinal data and extend those now to electronic health record-based data, which has even a third sort of structure, a particular way of looking at it. And this is a timeline of some plans coming up. We're going to begin year 3 in October of this year. This runs on the fiscal year cycle. And so we're just starting sort of the journey towards incorporating this new information, these new data sets, and using these methods and extending them. Next slide, please. And so question you might have is what's so different about EHR data compared to other types of what I'm thinking of as longitudinal data. And so longitudinal data for our purpose here is really using administrative claims data. And it's about complete capture of information during a particular point in a person's lifetime. And so here's an illustration where you can see different types of data streams about a person. So a person has recorded primary care physician visits. They have some dispensings they pick up at a pharmacy as a result of that. They may have a hospital visit and they have some diagnoses and procedures that happen during that hospital visit. All of that is recorded during a particular point in time. Now, there may be data that's missing. It's missing, though, on the whole, in the sense that the period of time is covered, but we don't have any laboratory results, for example, during that time, or we don't have any clinical vital signs during that time. So this is just the sort of concept of how is claims data actually coming through. When we compare that now to electronic healthcare record data, and particularly US-based electronic healthcare record data, we have a lot of detailed information within a single healthcare organization that maintains an EHR. But we may not have information about the same patient if they go to a second healthcare organization that operates a different EHR. And we may not know that that's the same person. And so we have this detailed look at one point in time for the patient, but it's probably much briefer. And we may have missing information outside of that healthcare organization. And so that's really the problem we're grappling with EHR data, is how to then bring this information into the signal detection sphere when, again, our real issue here is potentially lack of complete coverage of that patient during particular periods of time. Really, really critically in that is when we're looking to see, of all the people exposed to a particular drug, did they have this outcome? The concern here is that they had the outcome, but it was actually in a different healthcare organization and it was measured differently and it's beyond our reach. And so that's really the key point here of how do we deal with that in a signal detection framework. Next slide, please. And so want to spend 1 minute on the EHR challenges ahead and then turn it over to my colleague Josh, who works primarily with EHR data and can tell you all about it and will. But the idea here is, when we look at this, what I'm calling a standalone EHR data record, do we need to reach back to the techniques when we were using data that didn't have a denominator? Because we don't have this concept of complete capture of well-defined person time. Other things that we might want to look at, unstructured data is rich in potential available information, but it also may have lots of noise. So question about how do we filter that information? How do we prioritize it? How do we annotate it properly and ensure we then bring it back into a signal detection framework? And I want to note that in that same vein, there's been a lot of work looking at unstructured text in spontaneous reports. So just as there's unstructured text in the EHR that might be valuable, there's already been a lot of work that's been done in trying to leverage unstructured text out of spontaneous reports. So when we think about going forward, we want to make sure that we're synthesizing all of the prior work that's been done and take it and bring it forward. And so with that, I'm ready to turn it over to Josh to talk about the next challenges that we have using EHR data for the purposes of signal detection.
Great, thank you, Judy. So like Judy said, I'm going to talk to you a little bit about the challenges one might face. I did a project, you can go to the next slide. I did a project back in 2012. And so starting in 2008, researchers in Carol Friedman's lab at Columbia University, they showed that NLP could effectively identify diseases, symptoms, and adverse drug effects from unstructured EHR data. Later on, they used NLP to extract diseases and drug co-occurrence statistics from discharge summaries and also from Medline articles. Next. Yeah, but in 2009, building on that previous work, they used a collection of about 25,000 discharge summaries to extract disease symptom pairs and drug adverse effect pairs from clinical notes. So they filtered out certain sections. They didn't use, like, say, family history to try to look for correlations between those drugs and drug side effect. Next slide. That work inspired my actual master's thesis back in 2012. And that's kind of what I'm going to tell you about today, because I think that's a good example of some challenges and pitfalls that we're going to face as we're using unstructured EHR data. And also maybe we can talk about some opportunities to address those. It was basically a feasibility study on using NLP and clinical notes for hypothesis-free adverse drug effect discovery. Using natural language processing on admission note, history, and physical exam notes. We extracted the patient's current medications as well as their mentioned symptoms, findings, diseases, what we called clinical manifestations. The idea is basically that the H&P is a very feature-rich document listing all the patient's history and symptoms, even those that are unexplained. And we thought of that admission note as a snapshot in time. At that moment of admission, here were their current medications and their current clinical manifestations. Next slide. These drugs and clinical manifestations that we were extracting from the notes were defined using UMLS semantic types. So drug concepts included clinical drugs, antibiotics, pharmacologic substances. Clinical manifestations included a number of other semantic types. Mostly they were findings, signs and symptoms, and diseases and syndromes, among these others listed here. Next. We used a number of NLP tools, and I'll first talk about kind of the process of how we extracted that information using NLP, and then the results we found and what that tells us about what we're going to see going forward. The tools we used included the Knowledge Map Concept Indexer, which is it's used for concept recognition and negation. It's developed at Vanderbilt, but it's very similar to the NLM's MetaMap and how it works. SecTag is another Vanderbilt tool. We use it to identify section headers within clinical notes. Medex, a tool used to identify medications mentioned in the medication sections of notes, and RxNorm, we use their relationships that are from there to normalize clinical drugs to their ingredients. So this is just an example here of a clinical note, an admission note. So it starts out with name, MRN, date, there's a history of present illness. Miss so-and-so is an 80-year-old female with a past medical history of hypertension, congestive heart failure, etc. You also see a family history section and a medication section. You can go to the next slide. I have some nice slides that detail each step here, but I'm just going to skip right to the end for you guys. The yellow boxes indicate the different sections that we were able to identify. The green concepts like hypertension, CHF, hypothyroidism, dry cough, those are clinical concepts that we found, UMLS concepts, using the Knowledge Map Concept Identifier. The red ones are concepts that were negated. So it says the patient presents with a complaint of generalized weakness without fever, chills, and night sweats. Without is a negation word, and so the things after that are not symptoms that the patient has in this case. The family history section there, you see there's a blue line through that. We didn't use family history information because we wanted to look for drug adverse effect relationships or drug symptom, drug indication relationships for the individual patient, and we ignored family history in this case, which is not something we'll probably be doing going forward. Finally, the medications there, we identified different medication concepts, then we used, like I said, RxNorm to normalize those into their generic ingredients. Next. So given an H&P note, the procedure is as follows. We first extracted all of the patient's current medications and clinical manifestations that are mentioned in that note. That provides us with a list of drugs and a list of the current clinical manifestations. And then we take each pairwise combination of drug and CM, or clinical manifestation. And that's how we define co-occurrence, as the drug and the CM occurring in the same note. And we did know from previous work at our institution that that medication list in the admission notes was fairly accurate. Next slide. So we used 366,545 admission notes. And after we processed the notes, we extracted the drugs and clinical manifestations, and then we had a large number of drug clinical manifestation correlations to go through. Next slide. So whenever we used NLP to extract the clinical manifestations from our corpus, we did exclude certain sections and restrict by the semantic type of the concept. And for the medications, like I said, we normalized to the generic ingredients. But after that, for each drug finding pair, we calculated the odds ratio and the Pearson chi-square statistic. We used a Bonferroni correction to correct for multiple testing, which is probably an over-conservative correction. But when we also required the pairs to co-occur in at least 100 notes. Finally, we utilized a reference standard that was based on drug product labels and other sources to highlight the indications and known adverse drug effects. So you'll see the results here in a few minutes, highlighted in green for indications and red for known adverse effects. That was an automated system to kind of highlight those to help us focus on the other associations that we found. Next slide. Now, what you would ideally hope to find are some correlations that can be explained simply as known indications and known adverse effects, and then other strong correlations that could represent previously unknown adverse effects. Next slide. But what you actually find is significant confounding, often due to comorbid conditions and symptoms, making it very difficult to separate any potential adverse effect signal from the noise. But I'm getting ahead of myself, I guess. Next slide. We processed 366,000 admission H&Ps, 809,000 drug CM pairs is what we pulled out of that. That was about 1,700 distinct drugs, 10,000 distinct clinical manifestations. After we required, the minimum co-occurrence, so they had to occur together, mentioned together in 100 H&P notes, we were down to 75,000 drug CM pairs from 666 drugs and 2,100 distinct clinical manifestations. Next slide. After the Bonferroni correction, there were about 39,000 pairs that had significant chi-square. And based on our reference standard that we used, we were able to tag 10,500 of them as known adverse drug effects and 3,400 of those as known indications. So the accuracy of that 10,000 known adverse drug effects and 3,400 known indications does depend on how accurate our reference standard was. It was created using automated methods. Basically that still leaves a significant number of drug CM pairs that are not due to known adverse drug effects or indications. I'm going to show you here our selected results from this study. Like I said, this was done in 2012. Some of our NLP tools at the time were not as good as our current ones, but I'm going to show you the selected results from our top ranked correlations as well as from a few select drugs. Next slide. All right, so this is, like I said, this is just one example of our results data. We have, about 40,000 pairs that look like this over 600 drugs. And these are the top 50. This is about 25 on this page, I think. Next. So on the top, you see thyroxine and hypothyroidism strongly correlated. It's not surprising that they have a strong correlation since that is almost exclusively what it is used for. Next, Dornase alpha and tobramycin are both used in the treatment of cystic fibrosis patients. But note that Dornase alpha and tobramycin are also highly correlated here with pancreatic insufficiency. That's because pancreatic insufficiency is a complication of cystic fibrosis. So right here at the very top, this illustrates one of the major challenges. Comorbid conditions or symptoms act as confounders here, and drugs appear correctly correlated with their indications or adverse effects as you'd expect, but also with comorbid conditions of that indication or adverse effect. Next, just to point out a few more details here, aspirin, coronary arterial sclerosis are correlated with each other, which makes sense as an indication prophylaxis. Asthma and albuterol, as you might expect, are also correlated with one another. Next slide. Now you see here albuterol again. It's also correlated highly with exacerbation of asthma. So that's an artifact of the NLP in a way. It treats exacerbation of asthma as a completely separate concept than asthma, which in some ways it is, but in other ways it is, inherently connected. What the problem is with that is if you see exacerbation of asthma, say it's here 2,500 times, and on the last slide, asthma was there 9,500 times. Like maybe if they were mentioned separately in notes instead of together, the count would have been more increased, which can change how strong your correlation is. So that separation of concepts is something to be considered. Next. So it's not very informative, but it might be interesting to note that illicit drugs was highly correlated with abnormal bruising, and cocaine, as might be expected, was highly correlated with cocaine abuse. We also found much farther down the list that cocaine was significantly correlated with gunshot wound. So next slide. Aspirin and hypertensive disease were highly correlated again because of confounding, and statins and hypertensive disease were also correlated because of comorbid condition confounding. Next. Okay, so let's look at a single drug here. This is rofecoxib, or Vioxx, COX-2 selective NSAID that was on the market for over 5 years before it was withdrawn because of increased risk of myocardial infarction and stroke in 2004, I believe. So rofecoxib was indicated for rheumatoid arthritis, osteoarthritis, acute pain, and dysmenorrhea. And as you can see here, next please. We found it significantly correlated with arthritis, degenerative polyarthritis, and RA. It's also highly correlated with several cardiovascular symptoms, including myocardial infarction, which is the reason it was pulled from the market. So another important finding, these notes are with rofecoxib in the medication list were from before the drug was recalled, which means there was potentially a significant signal here at the time before it was known. But even if we were looking, it may have been lost in all the noise from all these other correlations that are present. Next slide. There we go. So prothrombin time increased. That's an interesting one. It might be related to the fact that Vioxx can have an effect on clotting time in patients taking warfarin. Next. So a few more correlations with rofecoxib. Pain and back pain, common indications are there. Other adverse effects listed include GERD, UTI, asthenia. Next. And it's also important to note here that stroke was another reason that it was pulled off the market, along with myocardial infarction. It did show up as a decently strong correlation there, but it's below our level of significance. So, but maybe whenever you're combining data from multiple sites and pooling them together, you would have seen a signal there for that. Next slide. This drug is rosiglitazone, or Avandia. It's used to treat type 2 diabetes. In 2004, 2007, there was a meta-analysis linking it to heart attacks, and it was withdrawn from the market in some countries or highly restricted in others after that. As you can see here, the indications are correct in the green at the top there. It's linked to diabetes. These three adverse effects at the bottom here, congestive heart failure, myocardial infarction, and anemia. Excuse me. Those seem to be correct because, we know that myocardial infarction was the reason it was pulled from the market, but also some later studies were unable to replicate that association. So this could definitely be due to confounding as well that we're not adjusting for here. Near the top, we see an association with hypertension. So there are studies showing that the drug lowers blood pressure in hypertensive patients, but hypertension may also be a comorbidity of type 2 diabetes. So it's unclear the nature of that correlation there. Next. Similarly, most of these possible adverse effects, these ones that we don't know what they are, obesity, along with obesity, hyperlipidemia, hypercholesterolemia, those all may also be associated with comorbidities of type 2 diabetes. Next slide. This is the one drug class we looked at. For everything else, we looked at individual drugs. But next. As you see here, most of these top correlations appear to be reasonable: hyperlipidemia, hypercholesterolemia, heart attack, stroke. And the same year that I did this study, statins were also linked to a possible increased risk of diabetes and memory loss. So I don't know. I mean, the diabetes thing could definitely be confounding. I don't know whatever came of that really, but on the next slide, yeah. So memory impairment, memory loss, memory observations, we did see correlations between those things. Memory impairment one was statistically significant. The memory loss and memory observations concepts that were pulled out of the notes were not significant, but possibly those were mentioned within the same notes or different notes and might have changed our counts if we, could find some way to normalize or aggregate those similar concepts that really mean the same thing. But that's a difficult question. Next slide. So our last example here of results is a simple example, insulin. And I think it's very illustrative of what we saw throughout. So as expected here in the green, you see it's highly correlated with expected indications, diabetes, hyperglycemia. The major adverse effect is there, hypoglycemia. And so it shows us, there's truth to be found in the text of these notes when you're mining them and looking at correlations. Next. What we also found is that insulin is highly correlated with a number of concepts that represent complications of diabetes. So you see here retinal diseases, diabetic neuropathy, nephropathy, kidney disease, diabetic foot ulcers. Next. And additionally, like rosiglitazone, insulin is highly correlated with many of the comorbidities of diabetes, including cardiovascular problems like CHF and hypertension, as well as obesity and hyperlipidemia. But importantly for our purposes, many of these concepts illustrate another point. If you'll click next. So retinal diseases, diabetic retinopathy, proliferative diabetic retinopathy. These are all different concepts that were found in clinical notes, may or may not have been from the same patient. Sometimes they mentioned diabetic retinopathy, and sometimes the concept retinal diseases was pulled out of the note, or proliferative diabetic retinopathy. Next. Here we see nephropathy. So diabetic nephropathy, kidney diseases, and chronic kidney insufficiency. Next. And then neuropathy concepts like diabetic neuropathy or just, plain neuropathy by itself. And then we also see foot ulcers and diabetic foot ulcers down at the bottom. And those neuropathic ulcers form as a result of the peripheral neuropathy. So it's important, I think, when you think about matching these concepts to a clinical vocabulary, there can be similar things that have different names or different concepts that are also very similar. And we just need to take that into account. Next slide. So what we found was that significantly correlated drug clinical manifestation pairs seemed reasonable, representing both known adverse drug effects and indications. We found that the correlations representing unrecognized adverse drug effects were potentially discoverable before they were known. And we also discovered some things like about how the NLP was sometimes coarse or ambiguous in nature. So agglomerating or, bringing those NLP concepts together to get a strong signal is difficult. And I think finally our main conclusion here was that confounding due to comorbid conditions and symptoms of diseases were very prevalent. Next slide. One of the key takeaways of this study that I did and other studies since then is that the vast amount of clinical narrative information in the EHR exacerbates the problem of confounding by introducing many, many conditions. So to ascertain causal relationships between a drug and an adverse drug reaction, confounders have to be identified. And adverse drug effect signals detected in the EHR are likely to be confounded by co-medication, confounded by indication, confounded by comorbidity, or any combination of those. Next slide. So since my early study, there's been a lot of work on how to address these problems. This paper here, for example, describes a method to eliminate confounding by comorbidity using penalized logistic regression. It takes into account not only confounders associations with the adverse drug reaction, but also confounders associations with the medication in question. And there are other published and data-driven regression-based methods that can also reduce confounding by co-medication and confounding by indication. So these problems that we encountered early on, there are multiple ways that people have proposed addressing them. Next. Another interesting example here, this study investigates whether spontaneous reporting system data can be used to adjust for confounding, and if it would result in more accurate ADR detection and the methods using SRSs or healthcare data alone. Next. I mean, so basically there's significant literature on exactly how best we can utilize NLP on unstructured text for adverse drug effect detection and discovery. And I think there's a number of ways we can tackle these problems. Next slide. I'll finish up here so that we have time for questions, but this is kind of a very incomplete list of questions that kind of came to me that we're going to need to answer and need to think about as we move forward. So for instance, which methods will we want to use to adjust for confounding? So should we limit ourselves to disproportionality analyses like that with FAIRS in my study, or should we focus on other study designs using the data? Questions, I think Judy mentioned, how do we annotate timing? So like we only looked at emission H&P notes as a snapshot in time for each individual patients. But as we move forward, we'll want to be able to say, okay, here on the timeline is the medication began, and here's later on the timeline where the adverse effects started. How do we deal about missingness in data? So we might have a lot of, like Judy said, detailed data for one single encounter, but then there's no encounters with that complete data for years after that. Also, how will we combine NLP extracted concepts with claims data? So for example, if you have an ICD code for hyperkalemia, and then you have a mention of the symptom hyperkalemia in text, and you also have an elevated serum potassium lab value showing the patient had hyperkalemia, are all those equivalent to one another? How do we treat them like with respect to one another? One of the things that came from my study, like I mentioned before, was how we can normalize those NLP extracted concepts so that we can see the true strength of signal. That might be more of a focus for the feature engineering core, but I do think we should work closely together to address that problem, because certainly a deep understanding of the underlying data is going to be necessary to determine the best methods here. Last, I was wondering, if we need a reference standard, so something that can identify indications and known adverse drug effects in a computable format. A lot of studies kind of create their own reference standard ad hoc. I've developed something that is like a reference standard that's computable, that's kind of automatically mined for multiple sources. Maybe we just want to use the product labels from FDA, but that's one of the questions too we're going to have to address. And again, like I said, these are only a few questions, and I'm sure everyone here will probably have more. And on behalf of Judy and myself, thank you very much. We'll be happy to answer some questions.
Judy and Josh, thanks for this really terrific presentation. We'll now open the question and answer portion of the webinar. And as a reminder, your lines are muted, so to ask a question, please type it in the Zoom question and answer feature. And a few people have already submitted questions, so I'll start with one here. I think this is more directed for Judy, by Antonio Ferreira. Are there any initiatives to try and have EHR data unified? In other words, having data elements and data format that do not vary so much between one EHR platform to another? If so, who is doing that, and where can we find details about such an initiative?
Sure, thanks. Yeah, I think that there's a lot of attempts using common data model concepts to put structured EHR data into the same format. So in the Sentinel system, we use a Sentinel common data model, but there is also the OMOP common data model. There is the PCORNet common data model. So there are many common data models that attempt to basically create the same structure amongst data so that data can be potentially combined or utilized together. I think the thing that also is sort of in its infancy is trying to use privacy preserving methods to be able to access data in the same structure at multiple sites without actually having to combine it. And so that that's a tricky one, because it requires knowing that patient A in site A is the same as patient A in site B. And that's a linkage concept that can either be done through direct linkage, which requires an exchange of PHI, or indirect linkage, which is a privacy preserving method, and that uses like a hashed identifier. And so there's a lot of work to try to do that, to create basically a better data system. So in thinking about attacking this problem of detection analytics, you can think about, do you pour your investment into improving your data systems, or do you try to get it on the back end and do it in your analysis step? And I think there's room for both in sort of thinking through this. Those are just some of the initiatives that are happening right now.
Thanks, Judy. Okay, another question here. This one's from David Carroll. Do you think structured claims data may have been helpful for addressing some of the confounding issues Josh mentioned in the NLP-based analysis of drug adverse event pairs?
Yeah, I do. I mean, I think we can. I mean, the data that's in the unstructured text is significant and meaningful, but the data in the claims data might have a little bit more truth to it in some respect, or we can be more certain because there's not a chance that the NLP got it wrong when it was extracting it. There's not that extra layer there. So I think that could be helpful. There's probably other ways we can use it that I'm not thinking of, but it certainly I think it certainly will be helpful having that claims data in addition to just the NLP for what we're doing here.
Okay, there's a lot of questions. We're not going to be able to get through all of them, but I'll ask a couple more. So this one's from Ramprasad Keshavamurthy. Does the Sentinel Innovation Center intend to develop new standard signal detection methods that will use labs and vitals in EHR data?
So I can take that maybe to start. I think, so the people you're looking at here on the video screen, so Shirley, Josh and myself are the Detection Analytics Core, and we're hoping to launch projects beginning in year 3 of the Innovation Center that are geared towards conquering some of these next challenges. And so I think one of the things we'll look at strongly is the utility of including labs and vital signs as part of the signal detection framework, knowing that they're primarily available in a subset of data that's EHR based. And so certainly I think there's a lot of room there to potentially add and enrich the signal detection environment, particularly as labs are more structured using link codes and other things like that. They're going to be more easy to incorporate potentially into future studies. I'll give it over to Josh or Shirley if they want to add anything as my co-core leads.
It's okay. That was a good answer.
Yep, I agree. Good answer. Okay, so next question. This one's from Yan Li. In Josh's example, what if the current medication list is incomplete? A patient may not report all taken medications in a given visit. Can we supplement that list with prior dispensing or prescribing information?
Yeah, absolutely. And I didn't go into the details here, but this the medication list from our admission notes is actually pulled in from multiple sources, including like the active medication list kept by providers through Vanderbilt. This was our old system before we adopted Epic a few years ago, so it may be different now. But in general, I think it's a good idea to pull in data from multiple sources. So the medication data used at Sentinel is very helpful, but I think there's a question as to whether or not it includes any information about over-the-counter medications. That's the kind of thing that would be in a medication list that you would get in an EHR from like a clinic visit.
Okay, I think we've got a few specific questions about details of the analysis. From Root Martins, did you do any work to map NLP concepts to a formal ontology to aggregate similar concepts?
I did, yes. So in this example here, I don't think this exploratory example here was kind of earlier in my work, and I didn't restrict it to a certain ontology. Later on, I've done work in like using SNOMED CT or possibly Medra for that. And I think Medra might be a good choice because it's pretty good in levels of like terms of hierarchy. And I think it would address some of these problems when you don't have a good restriction on vocabulary. I haven't done a lot in terms of concept normalization. And what I've been thinking about that is that, you know, there are certain levels of concepts. So you can be very detailed or you can get more granular. And I think there's probably different regulatory questions that you would be asking, where in some cases you'd want that really granular data. In some cases you'd want something broader. You know, so it's important, I think, to keep the granular data, but have some ability to move up and down the hierarchy when you're looking at like the frequency and co-occurrence of these concepts.
Another question from Anonymous is, how do you deal with rare events for rare diseases?
I mean, I think my answer to this, Judy could probably answer better, is that I think that's what we're hoping that we get out of Sentinel, right? Is that we're pooling our data from multiple places in hopes that we see these rare events frequently enough to find some sort of signal like that.
Yeah, I guess I would say that in that case, rare events from rare diseases actually is where FAIRS performs well, because there, if it's a rare disease and it's a rare event, it's very unusual. And so people may be motivated then to submit a case report or something like that. But one of the benefits of the size of the Sentinel system and the number of people covered, so, you know, we have PAD IDs, we have over 300 million. And so one of the advantages of such a large system is that if there's going to be any place where you're going to find the sample size for a rare exposure and a rare event, it's going to be in the largest system you can possibly have. And so that's one of the advantages of the Sentinel system, is its vast size.
So what are the biggest challenges that you see for signal detection in EHR data sources?
I think that confounding is probably the biggest one that we're going to need to address. Like you saw with my my little study there, like about more than half of our significant correlations were from some unknown source. So those weren't all new undiscovered adverse drug effects. A lot of them were confounding for different reasons. There are some different statistical methods looking at how we can address this. One thing I think that we have going for us is that the EHR data is kind of like everybody shows up at the hospital at some point. It's not biased in some ways. So we can use the data to maybe teach us what are the symptoms of disease or what are the comorbidities that occur with a certain ADR in question. So there's a lot of opportunity there as well, but I think comorbidities is probably the biggest thing. The NLP, I think there's been a lot of work in that area. And like I said, I think, smart ontology choices can maybe address some of those problems. Those are probably the two biggest ones in my mind.
Yeah, Josh, I'd add to that is and we've talked about it here, but I really think the timing issue is so important. I mean, one of the things that's so important for the FDA is to see that there's a temporal relationship and that the outcome is following exposure. And one of the things that's so hard to tease apart in unstructured data is what is exactly the sequence of timing of events when you're using notes or something like that. And how can we try to annotate that timing most effectively to make use of that information? I think that's really challenging.
You both make really great points. Following up on that, how transferable are disproportionality and signal detection methods with confounding adjustment that have been used in claims to EHR data?
I think it depends on the EHR data. I think it's quite transferable in EHR data that has some sense of real longitudinality. If you have really if you have an assurance that people are using the same healthcare organization for most of their care and things like that, when you have that kind of information, you have some assurance that you're not missing something. I think it's much trickier when you're looking at people who have their care spread out in many different places. It's fragmented, and you're not really certain where the missing information is. So I really think a lot of it is driven by the characteristics of the EHR system itself. And how well it's linked to the other pieces of data. Josh, I don't know if you had anything to add.
No, I totally agree. Like, for example, Vanderbilt's kind of a tertiary care center for a lot of people. They come one time because they're really sick or something. I mean, there's a group of people that come frequently too. So the data for those two groups of people is just very different, and one is very sparse and one is not. So completely agree.
Okay, final question. How do you envision the rich clinical data in EHR data sources enhancing Sentinel's signal detection capabilities?
I mean, so ICD codes have certainly been, useful tools for identifying adverse drug effects, but I think it's the level of granularity that you get out of EHR data. And I think that for things that are maybe difficult to associate with the drug, things where the physicians that are reporting to spontaneous reporting systems don't make that hypothesis connection in their head, I think it might be useful for finding those kind of things. Judy?
Yeah, I think one of the things to remember about administrative claims data or longitudinal data is that the things that make it into the data set are things that a physician is going to be, or provider is going to be reimbursed for putting that information in there. And if there's signs and symptoms or things like that that are important, but they aren't going to change the payment structure or anything like that, then there's just not that much incentive to make sure that it gets in the record. And that might not be true in the clinical record. In the EHR, you might see that information more readily. So I'm struck, in our current COVID, environment by, the signature right now for COVID is, losing your sense of smell. And so how often is that actually going to be coded? Because is there any incentive for reimbursement for somebody to code that you've lost your sense of smell? That's a kind of thing where it's really a clear connection. And the question is how often is that going to be available information? And it'd be an interesting study to do a comparison of claims in EHR and see, where is it more likely, and what's the correlation between that and reality. So just to think about, all data sources have their strengths and limitations, and so it's trying to find making the best use of the data that you have.
Thanks. I think we've addressed all the Q&A. Would you like to make any final comments?
Thank you, everyone, for coming.
Yeah, and I'd say we're looking for really great ideas. So part of the detection analytics core is to think about sort of where the next frontiers are, and where the next best methods are to develop and to sort of assist in terms of the FDA's mission of trying to find new and better ways to discover adverse events before, essentially back to Josh's point there, what could we have done if we go back in time and revisit rofecoxib, what could we have done earlier, finding stuff like that. So I encourage everybody to reach out and make sure that, we keep a community going in terms of the new methods and the new techniques.
All right. I would once again like to thank Judy and Josh for presenting on today's webinar. As a reminder, this session has been recorded and will be made available through Sentinel's webpage. You can also find details and registrations, links for future Innovation Center and method seminars on the website, sentinelinitiative.org. So this concludes our session, and we hope to see you again next time. Thank you.